Okay, well, I have to say I'm relieved I'm getting you all after the coffee break, so thank you for that slight change in the program. It's an honor to be talking about our attempts to develop a stem cell therapy for HIV/AIDS, and I'm speaking on behalf of both my own group and my collaborators at City of Hope, Dave and John. And with HIV/AIDS, since AIDS was first described, I think it was 32 years ago now in Los Angeles, we know we've come a long way. The sort of photographs we used to see of AIDS patients in the 1980s are now pretty much have gone away, and instead, because of the very powerful anti-HIV drugs that have been developed that are formulated into a single tablet these days, actually many AIDS patients are able to control their virus and live relatively normal lives. So this is great, but there's some buts. And one of the buts is that even in a wealthy country like this, only about 25% of HIV-infected individuals actually manage to access the drugs and have the drugs work well enough to keep their virus down at very low levels. And even for people who take the drugs and who control their virus, it's still really not perfect. Some of the problems that are encountered, there can be side effects. HIV-positive individuals have widespread inflammation throughout their bodies. They get diseases that can best be described as diseases of premature aging. There are toxicities associated with the drugs. Viruses sometimes develop resistance. And these drugs are hugely expensive. The estimate now for a newly diagnosed HIV-positive individual put on antiretrovirals is a lifetime cost of $500,000. And I think finally, and most importantly, no matter how well you take these drugs religiously every day, they never cure you. So you have to take these drugs for the rest of your life. So thinking, how could we do better than this? We actually took our inspiration from the 1%. And don't freak out, this isn't the part where we ask you guys for money. This is a different 1%. The 1% I'm talking about are the 1% of the world's population who have a mutation. It's a very innocuous mutation. They are absolutely normal, but they do not contain a functional copy of a gene called CCR5. And people who do not have CCR5 are profoundly resistant to HIV, and the reason is simple. It's because CCR5 is an important molecule that HIV needs to actually get inside our cells and infect them. So this idea that people who are CCR5 negative are resistant to HIV really was used in a very dramatic sort of proof of principle experiment, if you like, that was done with a single individual. Here he is. His name is Timothy Ray Brown. He spent many years living in San Francisco. He's also called the Berlin patient because he lived in Berlin as well for part of his life. And he's widely regarded as the first person who has ever been cured of HIV/AIDS. And the reason Timothy got cured was, first of all, because he got unlucky. He was HIV positive, and he actually developed leukemia. And his doctor at the time in Berlin realized that Timothy needed to be given a stem cell transplantation, a bone marrow transplantation, to cure his leukemia. And Timothy's doctor had the idea, maybe as well as trying to cure his leukemia and getting him a donor who is a tissue match, maybe we can find a donor who's also one of the 1%, and we can give Timothy a whole new stem cells and immune system that are CCR5 negative and are therefore going to be resistant to HIV. And that's actually what happened. Timothy, after his transplant, was cured not just of his leukemia, but also of his HIV. And so we would like to see if we can replicate what happened with Timothy, but not by giving HIV patients a bone marrow transplant, but instead by taking some of their own stem cells, their own hematopoietic stem cells, and engineering them to be CCR5 negative. In order to do that, we have been very lucky to have been funded by CIRM as part of a disease team. The title is Zinc Finger Nuclease-Based Stem Cell Therapy for AIDS, and it's a three-way collaboration My group at the University of Southern California, which is down in Los Angeles, for those of you who are local up here and may not know that. In addition, we do have representation from Northern California. We have a biotech partner, Sangamo Biosciences, based in Richmond. And in addition, also down in Los Angeles at City of Hope, we have our clinical partners. And the PI is actually Dr. John Zayer. So I'm going to show you one slide of data, that's all. First of all, introducing to you our technology. We use these reagents called zinc finger nucleases, and you can consider them to be sort of molecular scissors. They have the capability to go in and find a specific gene, in our case CCR5, and basically cut the gene so that it no longer works. And in this way, we are able to take hematopoietic stem cells out of a patient, treat them with the zinc finger nucleases, and make them CCR5 negative. And then the idea is we would then put those back into the patient. Will it work? Well, we have some proof of concept studies that we published whereby we took stem cells, human stem cells, either unmodified or treated with our zinc finger nucleases, and we put them into a special strain of mouse that we can actually then infect with HIV. And you'll see on the little graph down at the bottom, this is looking at the levels of HIV in the blood of these special mice. You'll see that the mice that received the unmodified human stem cells in black, their levels of HIV went up and they stayed high. However, for the mice that received the zinc finger nuclease treated the CCR5 negative stem cells, they were actually, although they did get infected by HIV, they were able to control it over time, and eventually the HIV disappeared. So it was very gratifying, of course, to publish that in the scientific literature, but even better, look at this. In December 2011, Discover magazine, doing their roundup of the top 100 science stories of 2011, decided that our little idea should come in at number 2. So we were so close. So, yeah. If anybody's interested, number 1 was that story nobody remembers now about the speed of neutrinos or something. So, whatever. So anyway, so this is the sort of the theory of what we would like to do. And with the money and the funding we got from CERN, what we've actually been able to do now is translate this to the clinic. And going from a mouse to a human, basically most of our issues are logistics. We have to figure out how to scale up the technology to work with the size of a human. In addition, we spent a lot of time making sure that the zinc finger nucleases work as efficiently as possible, and now with a single dose, we are able to knock out more than 50% of the CCR5 genes in the stem cells. And then finally, we've been performing a lot of preclinical safety studies that are appropriate for what would be a first-in-man clinical trial using these engineered stem cells. So then, just in one slide, this is showing you the progress over the last 4 years towards our clinical trial, starting in 2008 with the report of the cure of the Berlin patient, and then in 2009 and 2010, we were able to demonstrate that this could work using the zinc finger nucleases. CERN funded us as part of the first wave of disease team grants in 2010 for 4 years with $14.5 million. We have a little bit of it left if you guys want to give us some more. We've already been talking to the FDA and other regulatory agencies such as the RAC to get their feedback and their approval for our clinical trials. We're hoping to file our IND in the spring of 2014 and start actually treating patients by the end of the summer. So to summarize, here's my summary slide. Mother Nature, the 1%, and a very visionary doctor in Germany gave us the idea for how we could treat HIV and AIDS. We have a collaborative effort. It's a team. It involves academics, clinicians, and biotech partners, and that's what's needed, as well as the funding from CERN to move this to the clinic. We've quite honestly had a huge outpouring of support from patients and advocates. Really, it's humbling and surprising. And we realize that, for them, we need to do better than just offer them lifelong drug treatments. Our ultimate goal, if this works, will be to provide a one-shot, sort of safe, treatment whereby these engineered stem cells could be used by a patient to promote long-term drug-free remission from HIV disease. And then I'll just leave you with the last statement, which is, although we work on HIV/AIDS, because we're working with these stem cells and developing these technologies, we're hoping that they'll have a payoff with other diseases where stem cells could also be therapeutic. And I'll just leave you with my acknowledgement slide, the team of people at USC, Sangamo Biosciences, and City of Hope who've contributed this. And again, thank you so much. Okay.